biotechnology tools to understand bugs at a much deeper level than what we've done so far. So usually when we think about common diseases in calves are rotavirus, salmonella, crypto, E. coli, Coccidia pestis, and a limited number of other bugs that we usually, when we see scours, when we see pneumonia in an unusual way, we send those samples to the lab and we test for those. But what we are asking in this project is, one of the questions is it all? Using the new biotechnology tools and specifically sequencing directly from a sample without the need to grow any virus or bacteria, is there anything more there that we're not seeing? So we're using this new untargeted tool called metagenomics, where we go and test different samples, and we just look at all the genetic material that's in the sample, and then we look at those little sequences. We compare it with reference databases that have sequences of absolutely everything that has been sequenced until now. Could be a spider, a cricket, a whale, a fish, or bugs. So we compare each of those sequences to those reference databases, and it will tell us what's in the sample. And usually we find more stuff that we think that things that we've already studied and seen. So what are the questions of this study? We're testing healthy animals as well, because there might be a lot of bugs that are present in animals that are healthy, and because we don't test them, we don't know. So when we interpret what's in the sequence, we only test always the sequence. We want to know what's the background there. If there's anything else in the healthy ones that's background that's actually not causing anything, and we're over-interpreting the results in the sequence. We also want to understand the distribution. So we were able to secure funding to do this at a state level. So we were going to start with a few farms, and then we did an ARC linkage grant, and we got enough funding to do this at the whole state level. So we are going to include at least 60 farms in all the dairy regions, and we're going to take samples from, we've done 55 already, so by the end of the year we will be done with the complete sampling. And we're going to characterize the whole infectome, so all the microorganisms that are there in the calves that we sample. So that will be a very valuable industry-level information to understand what's in there and what's causing problems, and give us an idea of what's causing more problems. We also want to see if there's any specific practice associated with the presence of the bug. So we conduct a survey when we go into the farm. So the survey asks about colostrum management, housing, cleaning, use of vaccines, so things that we think might be related to the bugs that we find. And because we will also be sequencing all these bugs, we want to know if there are specific variants that are more severe than others. That's something that no one has looked at before. It's always, is rotavirus present or not? Is pestivirus present or not? But what's the specific genetic type? And because these viruses have been there for a while, there are different genetic types there. So we want to understand which cause more disease than others. So why is this important? We want to improve diagnostic. We want to make sure in the lab we have the specific battery of diagnostic targeting everything that's in there, not the traditional bugs that we know are present. It can help guide better treatments. So for example, do I need to use a certain antibiotic if the scours is caused by a virus, or something that doesn't really need antibiotic, or it needs some other type of control and treatment. And also to guide the next step for R&D. So if we find new viruses, if we find new bugs, then we need to find new tools to treat them. So now with the era of mRNA vaccines, for example, it would be relatively easy once we know the genetic structure of a virus to put it on a multivalent vaccine. So we're trying to add this in this P2 program, where we start unlocking the potential of the cow at a very early age. And we know that infectious diseases, scours and pneumonia are one of the main problems with calves. So again, the objective of the project is to obtain the complete range of microorganisms present at the whole state level, to compare these microorganisms between the healthy ones, to have an understanding of what's the background burden of bugs, and the sequence, and to understand those genetic variants. So what do we do when we visit a farm for this project? We sample 10 cows that are up to 10 days in milk, and the reason we sample cows is because we want to understand if the bags that are in the cows are in the calves. So not in all farms, but in most farms we try to get the cows. And we take nasal swab, rectal swab, and vaginal swabs. And from the cows, we look at 10 healthy cows that are up to 7 weeks old. And we take samples of all the ones that are sick, that have either scarring or have pneumonia, and we score them based on severity. And we do the survey where we collect information about colostrum management, housing, and other calf rearing practices. So in the lab, the samples, we culture them specifically for E. coli because we want to do a thorough study of the E. coli whole genome sequencing. But most importantly, we do the metagenomics part, which is we take this nasal swab, we take this rectal swab, we sequence absolutely everything that's there, which would be host, so bovine genetic material, there will be parasite, there will be viruses, bacteria, and then we compare it with this reference database to understand what's in there. And so these are the results from a pilot study. So before we embarked in this big statewide project, we did a pilot study with 2 farms. And we sampled 30 calves in each of these farms. They have a few animals with respiratory disease and a few animals with scarring. And we sequenced the samples. And what we found here, I'm going to try to describe because I know this can't be seen really well from the back. But these little circles here, these pie charts, they have information when we do the whole sequencing of we did the RNA, not the DNA, so we wanted to understand what was the RNA viruses first, and also what bacteria were doing more than which species of bacteria were there. So the DNA contains the same information in all our cells and in the bacteria, but the RNA is the way in which the cell expresses. So, for example, a cell from your eye would be different from a cell from your skin. They will be expressing different proteins. They all contain the same DNA, but they are different cells because they are doing different things, and that's what you capture when you do the RNA. Same with bacteria. Bacteria will be doing different things, and you can capture them through the RNA. And so when we did that, we found that usually this is the result from rectal swabs, and in these little circles, what you expect to see in rectal swabs is mostly an orange circle, which is bacteria. So you would expect that the whole circle is mostly bacteria. For some of the samples that had scours, we found that most of this is actually not orange, it's this gray, and this gray here is virus. And this is from a sample from calves that were 7 to 8 weeks old with watery diarrhea, and the virus that was mostly present was rotavirus, so nothing new yet. But then we did another sample with the scours of 43-day-old calves, and what we found is that it had a high content of virus, and this virus was the Toro virus, which is known to cause disease, but it's not really on the first differentials when a vet goes out there and try to understand what's causing the scarring. We also did the profile of the bacteria that were in there, and if you can see, like, if the shape of and the colors, these are the 2 that were scarring, and they have very different profiles in terms of the bacteria. So besides the rotavirus here, there's a lot of E. coli and there's a lot of Campylobacter. So the E. coli was not acting by itself. This usually have help to make the calf go sick. So that's some of the other stuff that we want to understand with this project. Similar with the respiratory profile. So what we found here, one of the interesting things, for example, here is that we found a virus called influenza D, which you haven't probably heard. This virus have been found everywhere where it's been looked for. We think it has a role in respiratory disease. In this case, we found it in healthy animals, but the respiratory role may come later in age. And that's something that we're trying to characterize as well. So we're finding all of this stuff that we thought at the beginning we will be looking at old stuff and new stuff. So bovine respiratory syncytial virus, we expect it to be there. But then there's a whole other range of viruses, picornaviruses, noroviruses, caliciviruses, that we're just starting to understand if they have a role or not in the disease process. So the other thing that we're looking at, for when I was telling you that we were looking at the RNA, is which of the genes of the bacteria are switched on when we have sick animals and unhealthy animals. For example, E. coli, if you look for E. coli in a fecal sample, you will find it all the time. But what if you target a specific gene that's switched on when the E. coli turns into a bad E. coli? That's the other thing that we wanted to test. And for example, these two red, the red highlights here, these belong to the two samples where there's cattle scouring. So what this is telling us is that the bacteria that are there are doing very different things in the cows that are healthy compared to the cows that are not healthy. And this is something that is shotgun, so we're just looking at everything, and it's telling us some really good guidelines into trying to see better diagnostic methods. We can also see the genes that are switched on in terms of antimicrobial resistance, and here what we found and what was expected, and it was nothing new, but tetracycline genes were very switched on in most of the farms. And the other thing that we're going to do is we're going to look in all the RNA viruses that we sequence, that are more than 20 different viruses that we have no idea what they do, but we will have an idea of how they transmit and how they evolve within the context of a statewide study, which is pretty cool. It's something that usually metagenomic doesn't meet epidemiology, but in this project we have the capability to do that, and to understand how the viruses are evolving and transmitting within cattle. And the reason I put rotavirus here is because we're not only looking at the new things, so for example this torovirus, or trying to look into viruses that haven't been tested for before, but also there's a story with viruses that are endemic, which is the variant story and the strain story, because we usually think about rotavirus present positive or negative, but we're not yet at the level of testing which strain of rotavirus. Is it the strain that we're using for vaccine, or is it a different strain? Is the strain that causing very severe disease the same strain that's in another farm that has always been present? And this rotavirus here that looked pretty common, this was a co-infection with two different types of rotavirus. And what some of these viruses do is they're like really doing like a Frankenstein thing, in which they recombine and they are more fit after co-infection. And this is another outcome that will be part of this project. So the data that we're getting is very rich in terms of everything that's telling us, and we're very excited that we're close now to get all our sample frame, and hopefully next year we'll have more information when we aggregate that data. But those are like not even terabytes of data. There's probably going to be petabytes of data that we have to go through before we have something that we can summarize. So thank you very much. That's it with the project, and yeah, I'm going to stop here. So we have time for the
let's thank Barbara. Do we have any questions for Barbara? Yes, Peter, do you have a big voice?
Yeah, I'll try. You've done a fantastic job there, but what about biomarkers within the immune system of the animal? Are you looking at those in conjunction? Or?
That's a very good question. And the more information you have, and it's something that I would absolutely love to get into, but then it gets so complex that it's really hard to interpret. We will have, for example, if you look at these pie charts here of the respiratory part, most of the genetic information that you get from a nasal swab is from the host, so it's bovine. So we can actually see here and try to do a shotgun to understand what's from the animal's marker, what's on the sick animal compared to the healthy animals. We will have the information, it's just the capability of analyzing it at the moment. But But yeah, that's a very good question. And that comes next, because this is big data, very complex, time consuming. But we hope, at one point, we hope we'll have everything together.
I also that.